Today I will talk about from the emergence of planktonic foraminifera to their present-day diversity. So, as you can see, the title is in two parts. It's because I'm going to do the presentation about two different work that are stemming from the same origin. So the first part, from the emergence of planktonic foraminifera, is based on the work that we have published last year. And the second part, to their present-day diversity, is a work that is in preparation since an extremely long time now about the global genetic diversity of planktonic foraminifera. So there's going to be a bit of disconnection between these two, but it stems from the same work of working on their diversity today. And none of the work that I'm going to present now would have been possible without all my co-authors from their discussion and all the help they have provided, especially with bioinformatics and the techniques to process large data sets. So when we talk about the present-day diversity of planktonic foraminifera, we cannot avoid to speak first about the diversity of foraminifera as a whole. So there are three large groups of foraminifera that are primarily benthic organisms, the monothalamids, lobothalamea, and tubothalamea. And planktonic foraminifera, in comparison, extremely not diversified. There is only 50 living species, compared to almost 10,000 species that are estimated to live in today's ocean. So there is really a lot of imbalance between the two groups. If we look into a brief overview of their evolutionary history, here you have a very extremely simplified graph showing their history. So first we have the emergence of the monothalamids that happens sometime during the Neoproterozoic, like any other lineage of eukaryotes. And during the Palaeozoic, you have the establishment of the main groups, the tubothalamea and the globothalamea, which then further diversified during the Mesozoic. And the planktonic foraminifera, they arrived quite late into the history of the group. They just appeared during the Jurassic. So the way we study diversity, or the way I do it, I'm just doing it from modern foraminifera because I'm working on the genetic, and the genetic is accessible only on recent foraminifera. So there are two ways of doing it. So you start from a plankton sample that you have recovered with a plankton net. So you can either select foraminifera one by one and isolate them into tubes where you are going to put also a buffer that is going to separate the nucleic acid from the shell. And from there, you are going to be able to produce sequences which are like tiny portion of the genome of the foraminifera, but that is informative enough in order to inform about the diversity of the group and their phylogeny. The second approach that you can do that is a bit more recent is that you can filtrate the entire sample. Then you extract the DNA of the whole community, and then you are going to have a strategy to sequence a shorter DNA sequence, but from all organisms that you have into your sample. That is going to give you access to a list of sequences that are raw sequences that you are going to simplify into a list of OTUs. This is just a way to simplify the data set in order to make it interpretable. But from there, from this list of sequences, you don't know to which organisms they belong. So what you do is that you produce a reference database from your barcoding data. This is true with foraminifera, but with any other kind of organism, because here your DNA sequence is linked to a taxonomy, hopefully. And from there, you are assigning your list of OTUs to your samples. So that's the way we do it. And what is very convenient when you work in molecular biology is that it's a world that is fundamentally digital, because you can, all information is encoded with only 4 letters, ATGC, which makes it extremely easy to standardize and to compare all your sources of data. So you have data repository where all genetic data that have been produced and published are available, freely available. And from there, you can also recover this metabarcoding data that have been growing in the last decades. So here, this is very famous studies from De Vargas et al. In 2015, where there was sample collected at 47 stations, 500 million reads. And then this data have been updated to a new version where more samples have been added and more sequencing. And the latest version is from 2022, where also sediment sample have been added. And in the time span of like 7 years, you have multiplied the amount of station analyzed by 10 and the amount of reads of data of sequences by 4. So what we have done is that we have gathered all data available, both from metabarcoding dataset and the barcoding data that we have for foraminifera into a single dataset that you see here. So the purple rounds are the single cell data. This is based on the work that has been done mostly by Kate Darling, Colombo de Vargas, and some other groups afterwards, including Bremen. Then you have all the metabarcoding data from surface ocean, which are the green triangles. There is also data from the aphotic zone. And then finally, we have less data on surface sediment samples. Okay, so we have an extremely large data set, but we cannot analyze it just like that. We need first to homogenize the data sources. So what I've been doing is to work on a series of scripts that would allow me to homogenize this data. And I'm not going to start to talk about the data in a natural order, because I'm going to talk first about what I've done with this third script, which is to first recover the metabarcoding, the foraminifera sequences from the metabarcoding dataset. For that, we use a database that is called the PR2 database, extremely used database. That is the protists ribosomal reference database. And basically, you are using it in order to separate the foraminifera sequences with all other eukaryotes that are occurring into your sample. Okay, as a first step, it works with like classical taxonomy. So the whole metabarcoding dataset is 250,000 of ASV. ASV stands for amplicon sequence variant. This is one single sequence that is representative for a type of organism that is occurring in your samples. And we have roughly 8,000 ASVs that are assigned to foraminifera into this dataset, and only 256 to the planktonic foraminifera. So the first thing that we have done is to look at the relative proportion of foraminifera into different environments. So we have the photic zone between 0 and 200 meters. Aphotic zone, everything below 200 meters, and then the seafloor, it's all the sequences that you found on your sample that are from sediment samples. What you see is that the foraminifera represent an extremely tiny fraction of the data set. It's between 0.001 to 0.01% in the photic zone. It's roughly 10 times more in the aphotic zone, and then between 0.1 and 1% on the seafloor. When you look at the relative proportion of foraminifera within the sample, you can see something interesting happening. So here we have 4 clades of planktonic foraminifera first, and then 4 clades. Well, the last one is unclassified, it's not a clade, it's just that we don't know what it is. But 4 groups of benthic foraminifera. And each graph here represents the different size fraction, because in metabarcoding sample, you need to size fraction everything. So you have everything from bulk sample, pico, nano, macro, and then the meso size fraction. This is the closest from what we are using in micropathology. And what you can see is that you have actually sequences of benthic foraminifera that are occurring into the photic zone, and it's not just by accident. So this is something that struck us when we were looking at this data, because if you have benthic foraminifera, they are supposed to be benthic. They are not supposed to occur in the plankton, because that's basically in the name. So what we did next, I'm sorry for the change in color code, it's just because it's from 2 different work, but it's basically the same data. So here what we looked at the propagation of planktonic and benthic clades into the plankton. So what we did is that we did a simple measurement of the proportion of each of these clades relative to distance from the coast from the sample station. And what we saw is that the benthic foraminifera from the clade Globotalamea is actually always present in the plankton, even if you are 1,000 kilometers away from the coast. So this is by summarizing all the data that you have, all the sequences that you have on one click, which is not refined because of course there are different species with different behavior. Then when we looked at is the dispersion of the ASVs or motus, this is equivalent here. And what we saw here is that for the monotalamea and the tubotalamea, you have a trend of decreased occurrences when you increase the distance from the coast, but not from the globotalamea that seems to have a similar behavior than with the spinos, non-spinos, and microperforates foraminifera. So what we thought afterwards is that then we want to know what is the identity of this globotalamea. So first, what we did is that we tried to separate a fraction of the globotalamea sequences that are actively dispersing in the plankton from those that don't. For that, we used a model to separate them, where basically we use the monotalamea and tubotalamea as being representative of real benthic foraminifera, and then the planktonic foraminifera to be the model for what is living always in the plankton. And then we separated the globotalamea in three groups, the pure benthic, pure planktonic, and those that we cannot purely assign. And then we try to map them on what we know is the diversity in the globotalamea. So we did this phylogeny, which is based on the work of Maria Holzmann. And we did a phylogenetic placement that allows us to kind of see what are the phylogenetic affinities of these two groups, those that are not dispersing and those that are dispersing actively. And what we can see is that there is no real differences between both, which tends to tell us that all globotalamea have the capacity to disperse in the plankton, even the textularis. And this is consistent with the occurrences of the Biophax genus in the North Sea that has been observed at John in 1987. So we see that actually the observations that we are producing here are not entirely new. So the next thing that we wanted to know is what is the actual affinities of planktonic foraminifera with the other benthic species. So we did another phylogeny where we put sequences of planktonic and benthic foraminifera together. And what you can see here is that you have 5 separate clades of planktonic foraminifera, which are the basal, non-spinos, monolamella, spinos, and microperforates. Sorry, it's 4, not 5. 5 different origin, but 4 different clades because the monolamella and spinos are always together. And what we ask as a question afterwards is to know what is the timing of divergence of this planktonic foraminifera from the benthic clades through time. So for that, you calibrate the phylogeny by using dates that you get from the fossil record. And we got 3 types of dates. First, the split between planktonic foraminifera species, so within the planktonic clades. Then a date at the base of the tree. So we estimate that the globotalamea emerged or separated from the texturates around 250 million years ago. And then one date to date the separation between 2 families of benthic foraminifera. And we did that in order to date at which point the different clade of planktonic foraminifera they actually separated from their benthic ancestor. Here we are not dating directly when they became planktonic, but just when they separated. And then we compared it to the actual history of the group known from the fossil record. So you have that curve that is redrawn from Lovery et al. 2020, where we see that the actual first forms of planktonic foraminifera emerged somewhere in the Jurassic. And the actual hypothesis about the origin of modern planktonic foraminifera is that they are all descending from one lineage that is called the Edbergella lineage. That is among the survivor of the chaotic crisis, where you see that here there is a huge dip in diversity that led to the establishment of the spinos and non-spinos foraminifera. Here, what the molecular clock suggests is that The separation of the different clades of planktonic foraminifera, so the spinos, non-spinos, microperforates, and others, is actually too young to be compatible to have separated from the benthics as the origin of the first Edbergella. So based on this result, we propose an alternative hypothesis about the planktonic foraminifera origin, is that you have a replacement of the Cretaceous planktonic lineages from multiple benthic ancestors after the KPG boundary. And we can support this hypothesis because the globotalamea have a widespread ability for dispersion in the plankton. It means that there is not a single origin, and you don't need to have a single origin for all of them because you have a community of globotalamea that are always dispersing in the plankton. We think that the establishment of planktonic foraminifera is linked to biomineralization because before the appearance of Roteabida or Robertinida, you don't have the possibility to do full biomineralization because all the globotalamea before are texturites, meaning that they need sediments to form their bodies, sediment particles. So when you have the appearance of actual biomineralization, it removes the need for the sediment, and then you can have a full life cycle into the plankton. And what we seek also is that the Cretaceous planktonic foraminifera likely do not have descending planktonic foraminifera lineages, and it's also supported by studies from the fossil record. And last, there is a continuous invasion of plankton by benthic foraminifera, even outside of crisis, and this is shown by the presence of Keliteria vivans and Dantiglobotalia infracta that have made that transition later during the Cenozoic. So now I'm going to go on the second part of the talk, where I'm going to talk about the modern planktonic foraminifera diversity, where we have somehow the clash of taxonomies. Why? Because from intensively studying surface sediment sample, there have been 50 morphospecies that have been described, starting with Alcide Bindi. And since the 90s, with the work of first work by Brian Huber, Colombo de Vargas, and then Cap Darling, there has been numerous cases that documented the presence of several cryptic species within morphospecies of foraminifera. So here you have the case of Orbuna universa, where the sequences of the SSU show that there was 3 clusters of sequences and that they were non-randomly distributed with latitude, suggesting that they would have ecological preferences. So that suggested that the actual morphotaxonomy would underestimate the true biological diversity in planktonic foraminifera. So what's happening is that these 2 lines of evidence, they are not necessarily in opposition, it's just that they don't record exactly the same thing. So what we need to think about is that the evolution is something that it's a phenomenon that is in perpetual motion. So here you have a theoretical case where you have a common ancestral morphospecies of foraminifera or anything else, which are composed of metapopulation. And while time is going on, you have this population that are going to evolve. They are going to eventually speciate and start to acquire different properties, either with the ecology or slight differences into their morphology. And if the time runs long enough, they are going to become separated morphospecies, which is actually what you observe into the fossil record. What we need to do is just to have a taxonomic system that is precise enough in order to capture this perpetual motion, basically. And that's what we have done with this pipeline. So I'm going to get back to it. So the first thing that we have done is to create all the Sanger sequences, all the sequences that we have from single specimen, because we can actually link them to morphospecies of foraminifera. So it's roughly 7,600 sequences. We retain only the sequences which are of the highest quality, which means that you observe them several times. And we produce a taxonomy using automated method of delimitation on these trees in order to have something that is objective. Then the second thing we did is that we assigned all the rest of the PFR2 database meaning all the sequences that you have not used to assign the taxonomy because they carry biogeographic information. Also, genotyping data that are techniques which are a bit old but produce lots of data. And also, we are using the sequencings from the metabarcoding dataset. And also, we try to estimate the diversity that is not covered by your reference database. And finally, we are going to look through a series of large patterns. So first, the global diversity, then the cryptic diversification, the latitudinal diversity gradient, and the effect of cryptic diversity on fossil assemblages. So that's the resulting molecular taxonomy. I'm going to spare you the details on how we get there. So basically, we have a 3-level taxonomic system. We have 41 morphospecies, which is the upper limit of your system. You have the 353 base types at the bottom. And in between, you have these 3 levels, which are the lineages, genotype, and base group that we are also calling motus level 1, 2, and 3. So I'm not going to get into details on that tree because there are better ones later. Then we merge this with the metabarcoding data. And this is what we have in terms of diversity. So you have 2 parts on the left graph. We have in total 45 morphospecies because we capture a few morphospecies that were not into our data set, which makes in total 94 genotypes. So it's just twice as much diversity as for the morphological diversity. And then you have the base group, ASVs, and base type, which is much higher. But here we are encountering a problem because what we know from the foraminifera is that they have a high intragenomic diversity. It means that sometimes every specimen can produce 20, 30, or 40 different sequences. And this is reflected into this rarefaction curve, where actually you see that this base type level, this diversity is kind of infinite, because if we want to capture the entire diversity there, it would mean that we have to sequence every single foraminifera in the world, which we are not going to do. But then when you look at the genotype and lineage level, which are representing the actual biological diversity, here we are saturating our diversity, which means that we have captured most of the diversity of foraminifera. Maybe we don't have everything, maybe we miss a few rare things, but the order of magnitude seems to be correct. The second thing that is interesting to note, here I put back this graph where we show the number of stations that have been sampled. This is the 2 things, the histogram in gray in the background. And I showed the cumulated amount of morphological species, lineage, genotype, and base group that have been formed through time, and also base type. And what you see is that through time, while the sampling is increasing, you have always an increase in the number of base types, ASV as well, but the number of genotypes that you find is saturating. And it's saturating even when we start the metabarcoding in 2010 and between 2010 and 2015, which means that most of the diversity that actually exists have been already covered with the single cell sequencing. Then we look into the major, let's say, axes that could fragment the diversity. So the geography, the size fraction, and the depths, these are upset plots. These are plots that are used to show the connectivity that you have between different categories into your data set. So to look at the geography, we separated the data into the Indo-Pacific, the warm Atlantic, southern cold water, Arctic, subarctic into the Atlantic and Pacific. And what we see is that the Indo-Pacific hosts most of the diversity with 80 taxa that are from there. This is only motus at the level 2. Then the warm Atlantic, and we see that the 2 are highly connected. Then they are connected with the southern cold waters. And in comparison, the Arctic, subarctic in the Atlantic, and the Pacific are rather disconnected from the other water masses. However, we see that in the size fraction, we don't see this level of disconnectivity, which means that basically you are finding all your taxa in all size fraction with a few exceptions that are negligible, and it's the same with the depth. This indicates that geography is your main factor that is structuring the fragmentation of diversity. Then we look at the data along phylogeny, where we can spend a bit more time, but I see I'm already at 30 minutes, I'm sorry. So I'm going a bit quicker now. And what we see is that you have three main patterns of cryptic diversification. The first is that you have no cryptic diversity in 29 species, morphospecies, and this is the most surprising. Then you have a cryptic diversification without habitat fragmentation, meaning that the cryptic species largely live in the same environment or the same thermal niche or in the same biogeographic area. And then a cryptic diversification with habitat fragmentation. And here this is the case for example, Buloides and Queen Coloba. And here this is the tree. This was the tree for the spinous foraminifera, and then here this is the tree with the non-spinous and the microperforate, and also the basal foraminifera, which are here. When we look into the biogeography, here you have maps with occurrence of the foraminifera. I showed here the example of Globigerinoides rubber albus, with here only 3 motifs level 2, and then you have 2 types of pattern. You have the cryptic species, the motifs that are globally distributed into the intertropical belt, or limited into the Indo-Pacific. And this is the 2 types of pattern that you find into the species that are living in low to mid-latitude. Then you have the case of the cold water morphospecies. So here is the case of Globigerina buloides. Here you have the entire morphospecies that is split into 2 lineages. One that is found in warm environment, and the other one that is found into the cold environment into the 2 hemispheres. And when you look into the details of how it's fragmented, you have either bipolar, meaning that you find them into the 2 hemispheres, or isolated in 1 hemisphere or 1 oceanic basin. And this is true also if you look at Neoglobochordina pachyderma. And here I want to draw your attention on the fact that in Neoglobochordina pachyderma, which is a dipolar species, you have actually 2 lineages that are occurring into the warm environment as well. When we look into the latitudinal diversity gradient, we can see what is the effect of cryptic diversity and how it's distributed. You have on the left the number of taxa that are found. So morphospecies, then number of motifs level 1 and 2. And then the curves with the dots, they show the intensity of the cryptic diversification. It's the number of motifs level 2 that and 1 that you find for each latitudinal bin. And you see that you have your kind of excess of diversity that are occurring between 30 and 45 degrees of latitude, which is a bit eccentric poleward compared to the morphological latitudinal diversity gradient. So finally, what is the effect on cryptic diversity on assemblages? So here I just took the ForSense database from Michael Sika and Kuchera, 2017, just to count the number of morphospecies that you have into the surface sediment. This is extremely classical pattern. And then I just calculated the proportion of morphospecies that are actually having cryptic diversity into these assemblages. And you see that it increases from the equator to roughly 45 degrees of latitude, where you are going to shift from 50% to 100% of your assemblages. And when you go further than that, basically all your assemblages is going to be composed of morphospecies that are going to have cryptic diversity. This tends to show that the primary driver for cryptic diversification is geography due to the difficulty to maintain gene flow over large distances. And despite we have all this cryptic diversity, the planktonic foraminifera remain under-diversified compared to the other planktonic groups. All right. that's the end of my presentation. I would like to thank you for your attention. Before we get to question, I want to make a quick advertisement about the FRAP principle. I guess you all heard about it. It's a series of principles to make your data findable, accessible, interoperable, and reusable. And none of the work I presented to you today would have been possible without this principle, because genetic data, they have to follow this principle. And all the things I've done is almost only on published data. So it would be very good if into the microbial community, we can attain the same degree of reusability. Thank you very much for your attention.
Well done. Very nice talk, Raphael. Sorry, just Helmer, would you like me to do the questions?
Yes, it'll be okay.
All right, okay, great. All right. Thanks so much. That was a really a wonderful talk. I learned about new things. Are there any questions from people in the audience? You can either write it quickly in the message, or you can just raise your hand and then I'll get to you. You can ask your question yourself, basically. So far, there's just been good comments. Thanks for the amazing and informative talk. Oh, this one. Hi, Lucas. Do you have a question?
Yeah, just because nobody else is asking one. Yeah, nice work, of course. We've talked about this already. I just, every time you talk about it, I discover new things that I don't understand that I need to ask about. And if we talk about this, or you showed these rarefaction curves, and that you showed it at one taxonomic level, I think it's the highest taxonomic level, we haven't reached or captured all the diversity yet here, especially the yellow. Yeah, the yellow and maybe one of the green ones. But on the next figure, you've shown that actually we've stopped discovering new discovery for a while.
Well, the thing here, it makes a plateau because actually we stop collecting forum, or we stop sequencing them, basically. Okay. I mean, it's just that at the beginning we were just sequencing single cell foraminifera. We continued from 2010 to 2015. It was basically me and Mathieu in the lab and a couple of students who were working on the sample that we were collecting. At some point, you just stop because every time you sequence a new forum, you find different sequences, but you don't find new cryptic species. And at some time, you just stop working because it's not worthwhile for you to produce again and again data, because they are just going to come up as different.
So this is the intragenomic variability within the species.
You're sampling the intragenomic variance. It's like if I would go around and sample people in the street, they would just all come up as being different.
So there's no secret place where diversity of foraminifera could still be hiding?
Well, you can never know, right? You cannot say that something doesn't exist. So there are a few species that we don't have. For example, we don't have Dalvernia primio. There are a few taxa that we did not get. So I know that the data set is not complete. But in terms of magnitude of the diversity, we are pretty certain that we attain like a real plateau.
Thanks.
Thanks, Lucas. There's a question from Gavin Foster as well. It says, fascinating talk. And why is the diversity so low in planktic foraminifera compared with benthic foraminifera?
Compared to the benthics, or compared to the rest of what is found in the plankton?
Maybe both. Start with the planktics.
Well, compared to the other group in the plankton, it's I don't have real explanation, and I think the reason is that they actually don't really speciate, and they don't really colonize a niche in particular. Actually, I think that they are rather they stay very generalist in the plankton, because and you see it mostly with the case of pachyderma that is actually occurring absolutely everywhere. Also, Globigernia uvula that is occurring in every single environment. they are not really limited by abiotic factor. Maybe they are limited because they are less successful than other foraminifera to reproduce in certain environments. But I think the reason is that actually they don't really speciate actively. And I think that the fact that we have cryptic diversity is largely due to how currents and the geography is organized in the world ocean. Compared to the other benthic foraminifera, what happens is that there is only one clade of benthic foraminifera that is producing planktonic foraminifera. These are the Globotolamea. So there is just the leakage from one clade, and also there is less potential niches into the plankton that you have into the benthos with deep sea coastal foraminifera, like the environment on coastal environments are highly structured, much more than in the plankton. I don't know if I answered the question.
I think so. I actually had a similar kind of question, because you've got dispersion going into the plankton realm, but so you can't have anything going back.
Yeah,
it's kind of a one-way street.
That's a one-way process, and somehow like the plankton is kind of a dead end for the benthic for the planktonic for the foraminifera at all, because once you're in the plankton, it's either you stay there or you die, but there is no coming back to the benthos.
Why is that? Because of the kind of specific requirements in a benthic habitat? Because when people culture forams, they're quite happy to live on the bottom of maybe not happy, but they survive for a while on the bottom of the vessel that they've been kept in, right? So
this I don't know, and we don't know either why foraminifera disappear completely from the ocean once you get on the shelf.
Okay.
It's really strange, but once you get into like close to the shelf, then you don't find planktonic foraminifera anymore, and this is kind of a mystery.
Okay, then there's another question from Sterios Janis. He was wondering if the findings are summarized in review paper, or if you have some references from the work that you just described.
I'm working on it.
All right, still working on it. All right, it's not published.
No,
okay.
it's taking very long because we tried to follow these FAIR principles, and in the FAIR principle, you have to produce the code that you have used to make all your analysis to make everything reproducible. So it's taking some time. Writing a review paper is long, and also I want to make the data readable easily by colleagues, and sometimes it's not easy to find the right balance and the right format for that. So I'm working on it.
Then there's a question from Susan Richardson. And she's wondering if any of the benthic taxa that you're finding in the plankton related to Planobelina or Triltomphalos, because she finds those attached to all this sargassum that's washing up on the beaches in Florida.
I have to check if that colleagues could send me an email with the names. I could check, but I don't have it in mind right now. Okay,
I'm sure Susan will follow up on that. And then there is a question from Tushar. Has single-cell genetic research in plankton foraminifera hit a dead end because almost all cryptic or genotype has been sequenced? I think you said that, didn't you? Or not?
No, because everything I showed today is only a pattern. We don't know about the process. And right now we have a glass ceiling above our head, which is sequencing the genome. And there is everything that has to do on the cellular biology, how they work, and what is their relationship with the other group of plankton that is linked to that. And this there is clearly a future. It's just that sequencing foraminifera is not sequencing. It's not necessarily challenging, but what is challenging is reconstructing the genome. And this is very difficult because if you have seen the pictures of living foraminifera, they are little universe that live in association with very different organisms. And when you sequence the single foraminifera, there is someone who has his mic open, and I would appreciate that they cut it. Yeah, when you sequence foraminifera, you are sequencing everything in association with them.
Yeah, cleaner bacteria. And so on. So Brian Huber just put a link, or not a link, but the title and details for a paper that was published in Nature Communications about the renewal of plankton foraminifera diversity. And so you can find that there. And then there's a question from Ri Ying. He says, what drives forum diversification in your view? For example, some species are more active than others, or what?
I think that I mean, we can speak about details, but what drives diversification is it's really the geography here. I show the case of Palaeozoic rubber. And here you have basically an axis that goes from the Indo-Pacific into the Atlantic. And what's happening in today's ocean is that it's separated in 3 parts. And here there is a geographic barrier that is harder to break than the other, because here you have a choke point. And it's kind of hard to have a gene flow that goes from the Indo-Pacific into the Atlantic. I mean, it's not hard to get into the Indo-Pacific, into the Atlantic, but once you are there, you cannot come back. So that's the first, and then the second is the north-south gradient, where when they hit a certain limit, they start to be overstretched, and then the gene flow cannot break the barrier, and then you just start to produce isolated genotypes. So to me, right now, it's really the primary factors that makes that you have cryptic diversification.
Okay, so would you suggest then during times when when we had a good flow through the equator, for example, the Cretaceous, that there would be less diversification because there was that kind of much easier going around? Or I mean, it doesn't really matter. It could be that in the poles there was less. I don't know.
Well, I would assume so. I would assume that, I mean, it's just an assumption, I cannot prove it, but I would assume that when the paleogeography is different, then the patterns of cryptic diversification are also absolutely different. It doesn't mean that it stops completely, but I would say that what we see today is really specific of how the quaternary climate functions.
Yeah. Okay. And then there's a comment from Malcolm Hart, and he says when he finds benthic species in the plankton, he seems to think that the ones that you find in the plankton are the same as you'd find underneath on the sediments, basically, and they're not kind of separate into consistent groups of taxa. So, but that's a comment, I think, is no question.
I need to read it because I did not get it.
Sure. Yes, the third from last comment.
Okay, I see. Yeah, I think so too, that it's what you find in the plankton is a sampling of what you find in the benthos, and it's not one group in particular.
Yeah. Okay. Cool. Any more questions? Please fire away.
Yeah,
Brian.
I'll ask one. Great talk, really exciting research. I find it very puzzling that in much of the plankton forum record, you really do have a lot of morphologic stasis. And at the same time, after the asteroid impact, you have extremely rapid speciation occurring, some of which is coming from the benthos, but we know Gumbolitra Cretaceae also is ancestor to the Danian lineages, very quickly being distributed globally. And I guess my puzzle is the idea of sort of a speciation mechanism. We know vicariance and all that, but at the same time, there's some pressure for this morphologic stasis. And I don't understand competition or incumbency among the plankton foraminifera. And so I wondered if you've thought about the other side of things, and that's kind of the longer term, maybe even 10 million year duration of some plankton forum species. And sort of why is that? They've got some ecological niche, they're happy there, I understand that. But then, when you take that away and it's something like the asteroid impact, then all bets are off and you have extremely rapid movement into these niches. So what's keeping species from speciating under sort of non-asteroid impact type scenarios?
That's a tough question. I think I would say I did ask myself the same question, not as detailed and put as eloquently as you did. But I think it's harder to speciate once you have an environment that is completely full of forms and different, not necessarily competitors, but you have less space to speciate, because my understanding of what's happening after the KPG boundary is that you have a surface of ocean that is virtually empty, or there is just a few survivors. If you think about, let's say, the Cambrian explosion, it's the same at the beginning. You have plenty of forms, extremely different plan of organization that coexist, and rapidly there is a filtration, and then you just have variation around a few of these plants. And maybe this is what's happening on planktonic foraminifera, because they stay in this morphological stasis, because what's happening is not that morphological species have cryptic diversities, that you have different lineages of foraminifera that at some point acquire a different morphology, and we don't, at least I don't know why. And it's true that we don't know what makes that you are going to have either a cladogenesis or an anagenesis. That is really not clear to me either. So I really cannot answer either.
Thank you.
Oh, and then there is another question from Kate Davis. Thank you, this is very neat. Could you comment on what you think may sustain bipolar distributions gene flow?
Well, that's interesting that you are the one who asked the question, Catherine, because you are among those who describe that actually planktonic foraminifera are not necessarily reproducing sexually. Actually, they are reproducing asexually a lot, which show that they don't have to rely on gene flow to actually perpetuate their own species. I think there is a moment where the gene flow is not maintained, and you have the cohesiveness of the species that is basically breaking apart the morphospecies. And you will see it more with, you see it a lot with Buloides. This is what you see here. And when Pachyderma, when you see it also with Pachyderma, which is more impressive, where the entire morphospecies is crumbled in several of these motifs. So what you see here is that in the bipolar, you are these two motifs here, the Globigerina Buloides 2A and 2B. You are the absolute limit of what they can do. But at the same time, and sorry, I think I misunderstood your question primarily. You see that they have ability to disperse because, for example, this genotype Pachyderma 1, it was found by Kate Darling only in the Arctic, and here you see that there are some sequences occurring into the North Pacific and also towards the South Atlantic. And this is what you see with the barcoding. I don't have it here, but I could show you also with Globigerines rubber, the pink version that is supposed to be only in the Atlantic, and we found it a bit into the Indian Ocean and in the Atlantic. So they can actually disperse, but they cannot establish a thriving population necessarily outside of kind of their what is the pool of their species. So there is actually a limit that we could calculate the maximum stretch they can afford.
That's really great, thanks. Any more questions? We've got probably time for another one or two. And if not, then we'll just again thank Raphael for a brilliant talk. Thanks very much. It was really informative. Yeah, it's great. Hopefully some of you, if you're going to forums, Raphael will be there as well, so you can have a chat with him there if you want as well. And quite a few of us might be going there, so see you all there. And just to remind you that next month will be Annika Brombacher who will give a presentation, basically. So look forward to seeing you then. And a nice continuation of the day.